All right. Okay, come on, lecture capture. Let's wait just a minute to see if the lecture captures this. Ah, there it does. It just came on. Great. Okay, so the quiz, as far as I know, just opened. And so obviously some of your classmates have chosen to perhaps find a quiet place and take the quiz during this hour, supposed hour of scheduled class time, which is why I'm lecture capturing this today. All right. So any questions? I don't really have any additional announcements. I think this has been a big week for announcements, but most of the events and things that I was telling you about have already happened, except for the fact that I have canceled lecture for Monday, but you do have discussion section, okay? I think everybody's clear about that. I've just been neglecting to put that in a Canvas announcement, so, but y'all are aware of that. All right, so in case you were wondering why I spent about 10 minutes last time talking about the kidney, there are a couple of take-home lessons from thinking about the embryology or the development of the kidney. And one of the first things to remember, one of the first take-home lessons regarding the kidneys in a developing fetus is that the developing kidneys are incredibly closely aligned with the maturation or the development of the gonads, both the ovaries and the testes. In fact, some of the ducts that we see, and I'm just pointing these out, the metanephric duct and the mesonephric duct. First of all, the mesonephric duct is firstly a kidney draining type of tubule, but then it becomes the vas deferens or the Wolffian duct in a developing male. Okay, so we see this. We see that the duct, especially the ducts, and certainly the close proximity of the developing kidney and the developing gonads just is an interesting sort of developmental stages that we see in a developing mammal and in developing humans as well. So I did describe a little bit of comparative anatomy, never hurt anybody. We were looking at comparing the kidney development in other vertebrates, so we weren't talking about insects or other kinds of invertebrates, but vertebrates that we assume are more closely related to each other than most of the invertebrate groups. And so the number one take home, how closely related the development of the kidney is to the development of gonads, both ovaries and testes. But secondly, how interesting it is that when we look at the development of the mammalian kidney, that we see we're essentially looking at evolution. We're looking at the transition from simple, mostly aquatic organisms, the fish and the amphibians, and then we are looking at the transition of those kinds of animals to the more terrestrial animals. So this is a really fun way to think about some of the evolutionary changes that have happened in the evolution of mammals that show through development some of these evolutionary stages. Okay. So having talked about the gonadal development, and I hope making a good case for the similarity in development between the ovaries, the testes, and the kidneys, probably most of you all remember that very early on, in fact, up up until about 6 weeks in a developing human embryo, there is no differentiation between males and females, okay? Now, we know that in mammals, the sex is determined by chromosomes, more specifically, some of the genes on the chromosomes, and we will be talking about that a little bit later in the course. But up until 6 weeks or so, the gonadal differentiation in humans does not exist. But you may notice some of these, some of the labels here are the same as we see in the developing kidney. So the mesonephric duct, for instance, mesonephric tubules. All right, so, but at between about 6 and 7 weeks of fetal development, what we see is now there is some differentiation going on. And so males are on a track to form testes and the external genitalia that we associate with males. Females are conversely on a track to develop ovaries and, again, the external genitalia that we associate with the female sex. All right, so, but what happens is, because this is an undifferentiated stage up to 6 weeks, we have both. We have both the potential for male and female. In other words, we have all that duct work that we associate with being male, and the vas deferens, for instance, that carry the sperm to the point of ejaculation. We associate the fallopian tubes with capturing the ova that are released from the ovary. So some of that quite possibly and quite definitely was originally associated with the kidneys, okay? So just another reminder of the similarities that we see here in mammals between kidney development and gonad development. Okay, so what happens, so here we have the undifferentiated, so we have the Mullerian duct here in red, and we have the Wolffian duct here in blue, and so up to 6 weeks we have both in the developing fetus or embryo, and then they start to degenerate, depending on whether this is going to eventually be a female or a male. So in the male, the mesonephric duct persists, and then we start calling it the Wolffian duct, okay, or the vas deferens. And the other, the Mullerian duct, just degenerates, okay? But it is retained in the female. So the female retains the Mullerian duct, which becomes the fallopian tubes or the uterine tubes, okay? So notice in both cases, we essentially develop two sets of tubules, ducts, one to carry urine to the bladder from the kidneys, and the other to carry whatever the gametes are, whether they are sperm or eggs, to the point of ejaculation in the case of the male, or fertilization in the case of the female, okay? So I am not going to be, I'm not going to put up a diagram and have you label all this thing. I just want to have you all have an intuitive idea of what's going on here. And technically what's going on very early in embryonic development. So like I said, I'm pretty sure that some of your basic biology courses, maybe in high school, talked about this, undifferentiated until 6 weeks. And then following that, usually through the intervention of the SRY region on the Y chromosome, the sex-determining region of the Y chromosome, it kicks off a cascade of hormonal responses that cause the developing fetus to become a male, okay? So we'll talk about some things that can happen. So I've mentioned before, what happens if the SRY gene is transposed to the X chromosome? What would happen? I mean, those are things to think about. And we've got a pretty good, very simple to read article that describes what can happen. That sex is not as clear cut as some people would claim it is. Okay, we can have chromosomal sex, but then external appearance can be completely different. All right, so just keep in mind flexibility of sex. All right, oops, I wasn't going to show you that yet because I was going to propose another comparative vertebrate scenario to you. So let's think about reptiles now. Are there reptiles that spend a lot of time underwater? Yes or no? Yes, yes. We have some here on campus, right? The turtle pond, yes, the turtle pond. Crocodiles, alligators, these reptiles are known for going underwater and spending a lot of time underwater. Now, they are perfectly decent vertebrates, so they have to breathe, but they can stay underwater for a really, really long period of time. Because what happens to their circulatory system is that their circulatory system and their heart continues to circulate blood through their body, but it bypasses their lungs. Are lungs really any use if you're underwater? Not really. There's some residual oxygen in there, but there's also residual oxygen in the circulatory system. So these animals it's an autonomic response. They don't have to think about it or adjust their physiology somehow. It's just underwater. They just have some anatomical means of bypassing their lungs. Now, they're not bypassing it completely, right? But keeping the oxygenated blood circulating. They can't stay underwater forever. They're not fish. They're not extracting oxygen from the water, although there is some evidence that turtles can do that with their anuses. I'll let you think about that over the weekend. But they can't sustain themselves like a fish underwater, okay? They eventually have to come up for air. But this gives them a really long period of time that they can stay underwater. So my question to you is, are there ever instances in mammalian life in which the same thing happens? That a mammal is surrounded by liquid and is sustained in a liquid environment. Dolphins, yeah. What about, that's true. They do have to come up, though, periodically for air, the blowhole. You mean like a baby? I mean, yeah, like a mammalian fetus. Yeah, so, the fact that we're talking about prenatal development, big hint, right? Okay, yeah, this is something that is a really interesting phenomenon that we see in prenatal mammals, including humans, of course. Where is a prenatal mammal, a prenatal human, embryo or fetus, where are they getting their oxygen, their nutrients, et cetera, et cetera? Where are they getting oxygen and depositing carbon dioxide? Where does that exchange take place? From the mother. I heard the word barrier. What barrier? Placental barrier. Exactly. So what good are their lungs? Not for exchanging gases, right? And so in prenatal development, what we see, and it is depicted right here, so they're using the regular colors of red and blue or purple to indicate oxygenated red blood versus deoxygenated blue blood. And so they're picking up oxygen here in the placenta. So we're just going to talk about oxygen here. And the placenta then sends this oxygenated blood out into circulation in this developing fetus. All right, it gets to the heart, and then it is sent out of the heart, bypasses the lungs in a blood vessel called the ductus arteriosus. Ductus arteriosus. This is found in all, this part of a circulatory system is found in all vertebrates, but it looks really different when we're looking at it in a fish or an amphibian. We're looking at mammals now, specifically humans. Okay, so the blood comes in, it gets pumped past the lungs. Now, some has to go to the lungs because the lungs are developing, right? So they need oxygen. So you can't just bypass those developing organs completely. But as far as usefulness in picking up oxygen, the lungs are not useful at all. Okay, only the placenta is responsible for gas exchange, nutrient exchange, which includes waste products and carbon dioxide. Okay, so think about what has to happen immediately upon birth for a mammal. They have to go from this completely aquatic environment with dependency on the placenta and this called the umbilical vein, and to a situation where the placenta is lost immediately. The lungs have to dispel or get rid of the liquid that's in the lungs, and they have to start functioning. Okay? They have to start functioning. Now, you remember when we were talking about stabilizing selection and the fact that big babies are born, usually premature, because they are so big and they just trigger childbirth, but then their lungs are not mature enough. So it does take a certain amount of time prenatally for lungs to develop the capability of being able to function immediately upon birth. All right, now, I told you that crocodiles and turtles have a way to shunt blood away from the lungs and keep it in circulation. Prenatal mammals can do the same thing, and they do it through this portion of the two atria of the heart. So there's essentially a connection or an opening between the two atria in a prenatal developing fetus. It's called the foramen ovale, okay, the foramen ovale. So immediately or very soon after birth, we see that the foramen ovale closes up. We lose this placental, the placental veins and vessels. And then in the adult circulation, we still have a remnant in the form of a ligament that used to be the ductus arteriosus. And we have this ligament that used to be the ductus venosus, but are no longer functional. Okay? They're no longer, they're just ligaments. They're not sending blood. They're not part of the circulatory system anymore. Okay? So, this is an amazing physiological feat that mammals have to undergo in order to go, again, from that aqueous environment to breathing air and using lungs then. The lungs come online, essentially. So circulation through the foramen ovale, right after birth, so it's kind of like a flap that closes up because there's a pressure differential between the two atria. So it essentially closes the foramen ovale, and for most people, eventually that is sealed. okay, it becomes solid. So pressure differentials between the atria cause the foramen ovale to close almost immediately after birth. And then the fusion takes place during infancy. However, in approximately 25% of the population, the foramen ovale does not seal properly. This is not a heart murmur, okay? Heart murmurs are associated with the valves, okay? But this is known as patent foramen ovale, and most patients don't even know they have that condition, okay? I have the word, those of y'all who are going to go into the health sciences, you will see this word a lot, patent, and that's how you pronounce it. It's not patent like, you know, you get a patent for an invention or something. Patent means open or unobstructed, affording free passage. For example, the bowel may be patent as opposed to obstructed. So you see that that's a medical term that you can see pretty often. So I'm going to show a video that addresses this incredible topic of the foramen ovale. I'm sure we're going to see a commercial, maybe. Or not. Yay. We'll do close
Fetal
down.
Circulation by Lisa McCabe.
Hello, my name is Lisa McCabe. I'm a clinical nurse specialist at Children's Hospital Boston in the cardiovascular program. We are now going to talk about fetal circulation and the transition to postnatal circulation that occurs after birth. The specific structures associated with fetal circulation include the placenta, the umbilical vein, the ductus venosus, the foramen ovale, the ductus arteriosus, and the umbilical arteries. Blood rich in nutrients and oxygen supplied via the placenta flows through the umbilical vein to the ductus venosus. Blood flows from the ductus venosus into the inferior vena cava up to the right atrium. This blood mixes with blood returning to the heart from the upper body via the superior vena cava and from the lower body via the inferior vena cava. Once in the right atrium, some of the blood flows to the right ventricle and some of the blood flows through the foramen ovale to the left atrium and into the left ventricle, where it is then pumped out into the aorta to the body.
Point of clarification. The flow of blood from the right atrium to the left atrium via the foramen ovale enables oxygenated blood from the placenta to reach the coronary arteries and cerebral circulation, the two most metabolic active organs in the developing fetus.
Blood that flows into the right ventricle is then pumped into the pulmonary artery. Because the lungs are fluid-filled instead of air-filled, the vessels in the lungs are narrow, creating higher resistance to blood flow into the lungs. Due to this high resistance to blood flow in the pulmonary circulation and the low resistance to blood flow in the systemic circulation, blood pumped into the pulmonary artery by the right ventricle is more likely to flow into the ductus arteriosus and then into the aorta. Due to the high blood flow from the superior vena cava and the inferior vena cava up to the right atrium, pressures in the right atrium are higher than in the left atrium. This promotes blood flow through the foramen ovale at the atrial level. Only 8% of right ventricular output flows into the lungs, providing nutrients for the developing lung tissues. Most of the blood passes through the ductus arteriosus into the aorta and out to the body. The umbilical arteries allow blood to flow from the body back to the placenta to be enriched with oxygen and nutrients. Once the baby is born and takes a breath, the umbilical cord is clamped and the placenta is removed from the systemic circulation. Immediately, the transition from fetal to postnatal circulation begins. With the elimination of the placenta from the circulation, systemic vascular resistance begins to rise. With each breath, more alveoli in the lungs expand and the vessels surrounding them dilate in response to the presence of oxygen. Pulmonary pressures begin to decrease. Although pulmonary pressures are lower than systemic pressures within minutes after birth, it is 6 to 8 weeks before pulmonary vascular resistance decreases to normal. In the postnatal circulation, blood no longer flows through the foramen ovale or the ductus arteriosus. The increase in left atrial pressure after birth forces the septum primum against the septum secundum, functionally closing the foramen ovale. Within 3 months, the foramen ovale permanently closes as fibrin deposits fuse the layers of septal wall together.
Point of clarification. While the patent foramen ovale, or PFO, may close by this time, it also may not. Autopsy studies in adults indicate that 15 to 25% of adults have a patent foramen ovale, which never close. The shunt via the PFO after birth is usually small.
The ductus arteriosus begins to close shortly after birth, once the infant begins to breathe. Normally, the ductus closes completely in 4 to 10 days. The ductus venosus is open at the time of birth, making central venous access possible through the umbilical vein. As fibrin infiltrates the ductus venosus, it usually closes within 3 to 7 days. After it closes, the remnant is known as ligamentum venosum. Within a week after birth, the umbilical vein and umbilical arteries are infiltrated with fibrin and also become ligaments. Thank you for your time. This concludes our lecture on fetal circulation.
Please help us improve the content by providing us with some feedback.
Okay, so I think she does a really good job of telling you or explaining with the little cartoons that she has of what is going on and then what happens at the moment of birth. And so there are several immediate physiological changes that happen when a mammal, including humans, are born. They mostly are associated with the cardiovascular system as well as the respiratory system, okay? So this just gives you some, just a reference to the fetal circulation. So this basically just is a list of what she was talking about in that video. Loss of umbilical circulation to the placenta, closure of the ductus venosus, closure of the foramen ovale, closure of the ductus arteriosus. So these three structures, two of them are ducts, one of them is the foramen. So the word foramen means opening, okay? The foramen magnum, do y'all know where that is? Can you point to it? A little further back. Yeah, it's the opening to your skull. Okay, so foramen is just a anatomical word that you probably should pay attention to. Large increase in pulmonary circulation, sure, because now essentially the lungs are coming online, they're becoming useful, they're being set up for actual gas exchange, which they were not before. An increase in renal blood flow from a decrease, renal vascular resistance, and then the change in skin blood flow as well. And then as far as the respiratory system, loss of placental gas exchange, okay, that makes sense. Initiation of ventilation of the newborn's lungs, commencement of pulmonary gas exchange, and the establishment of a functional renal capacity. Okay, so if you want a little bit more information about physiological changes at birth, here are, it's put in a nice tabular form here for you. They didn't spell foramen correctly, but these are the three main structures that we find in a prenatal mammal and that get changed after birth. And sometimes it takes a few months, sometimes it occurs almost immediately. So, for instance, the ductus venosus and the foramen ovale functionally stop working within just a few minutes of birth. Okay, so, and then there is the eventual switching, and she mentioned that it's the incorporation of fibrin that causes these ducts to change into the ligaments. Okay, the ligamentum arteriosum, fossa ovalis, and the ligamentum venosum. Okay, so the reason that so this idea that you can keep oxygen in your circulatory system and you can keep circulating oxygen through your body until all the oxygen is depleted. I hope that a lot of you have taken CPR lessons or you are familiar with CPR. If you remember, and I used to have to demonstrate this every year, as over my lifetime, they've gone from, okay, let's do like 5 compressions to keep the blood circulating and then a breath. And I think the ratio now is 30 compressions to 1 breath, because it's way more important if somebody's heart has stopped to keep the circulation going rather than replenish the oxygen in their lungs. And I used to think, you know, hey, you're blowing like carbon dioxide into this person. And the answer is no, you're not really, because most of your exhale is also oxygen, okay? All right, so any questions about that I might be able to answer? I'm not as educated medically as the nurse practitioner was in the video. But this isn't, you know, it's, I think we forget sometimes, or we get used to just thinking about childbirth or mammalian birth in general. And sometimes we forget what an amazing physiological transition that is from, again, like I've said several times, from this aqueous environment, and you're essentially like scuba diving, to being able to breathe atmospheric oxygen, okay? And if I ever teach comparative vertebrate anatomy again, it's an amazing evolutionary transition from fish through lungfish, which can actually use both lungs and gills, to terrestrial animals. It's a really, really fascinating transition evolutionarily, okay? So let's go back to thinking about prenatal development. And as I've said, every class period for the last week and a half, lots of things can go wrong, very few things do go wrong, but we should address some of the aspects of prenatal development that can go wrong. And one of the main problems associated with abnormal fetal development are the presence of teratogens. Teratogen is a general term that means it's a substance that may produce physical or functional defects in the human embryo or fetus after the pregnant woman is exposed to the substance. So I'm pretty sure a lot of you can come up with what would be potential teratogens. And this is not a course on teratogens, but I am going to address some of the major teratogens, some of the ones that we either can't control because they're in our environment, or ones that are within our control. So this is not a teratogen. Okay, pop quiz. Let's just take a look at this.
Some well-known harmful teratogens are viruses, such as the flu or rubella, alcohol, nicotine, prescription drugs, such as antidepressants, or radiation. It's interesting that all of these are particularly dangerous during certain periods of prenatal development. Viruses have a major impact early on in pregnancy, but not so much towards the end. Specifically, if a mom gets the flu between month 4 and 6 of pregnancy, the risk of schizophrenia increases for the child later in life. Now, the odds only go from about 1% to 4%, but it's a significant increase. In addition, nicotine exposure from maternal smoking can lead to low birth weight or even stillbirth. So this shows that the developing brain is really vulnerable to both environmental stimulation and toxic chemicals. That's why prenatal care is so important to a developing fetus.
Okay. All right. So teratogenic agents come in a variety of forms. They can be chemical, physical, biological, but anything that produces abnormalities during embryonic or fetal development is called a teratogen. Okay, so there's lots of different things that can affect a developing fetus. Maternal disease, altered metabolic state also can affect the development of the embryo or fetus. So theoretically, teratogenic agents can cause birth defects in 3 different ways: by direct exposure of the pregnant woman and the embryo or fetus to the agent. So for instance, something like alcohol. Remember my rule of thumb, anything that can affect your brain. They mentioned some drugs, like drugs for depression. They are psychotropic drugs. They are capable of crossing the blood-brain barrier to help therapeutically people who have depression. So anything like alcohol, like nicotine, anything that can pass the blood-brain barrier, chances are excellent it can also pass through the placenta. So direct exposure through exposure of the soon-to-be pregnant woman to an agent that has a slow clearance rate, such that a teratogenic dose is retained during early pregnancy. So something that a drug, let's say, that is fat soluble, like tetrahydrocannabinol, THC, the operative agent in marijuana, accumulates in the fatty tissues of the body. And so it is retained for much longer than any kind of drug that is water-based or water-soluble, okay? That is good to know if you're applying for a job and you just went to a party on Saturday night and you're going to have a drug test on Monday. Chances are pretty good that THC could still be in your body and detectable, even if you're not getting the psychotropic effects of THC. Okay, that's my public service announcement for the day, for y'all. Okay, as a result of, so here's another way that teratogens can affect mutagenetic effects of an environmental agent that occur before pregnancy, causing permanent damage to a woman's or a man's reproductive cells. So something like radiation that might affect gametogenesis in either females or males can be a source of a teratogenic agent. Okay, so we tend to think about mothers, pregnant people consuming something that is going to cause damage to a developing embryo or fetus. We cannot forget that male exposure to some of these teratogenic agents can also affect sperm development and perhaps even change DNA methylation patterns in the spermatogenesis, okay? So factors influencing, and I know we've got a lot of text here, but a lot of that is because we don't have a textbook in this course. Several considerations in determining the type and the amount of damage that might result from exposure to these particular teratogens. These include timing of the exposure. Structures in the body are vulnerable to the most severe damage when they are forming. And not all organs within a developing fetus are developing at the same rate or at the same time, okay? If a substance is introduced during a particular structure's critical period, that is the time that it's most critical during development, the damage to that structure may be greater than if the exposure occurred at a different time that would not affect that particular growing organ. For example, ears and arms reach their critical periods at about 6 weeks after conception. If a mother exposes an embryo to certain substances during that period, the arms and the ears may be malformed, but once they are on a track to normal development, if a mother has exposed to some kind of teratogen, chances are that might not affect the ears and the arms, okay? The amount of exposure, certainly, you could probably come up with that yourself. Some substances are not harmful unless the amounts reach a certain level. Critical level depends on the size and the metabolism of the mother. The number of teratogens, so fetuses exposed to multiple teratogens typically have more problems than those exposed to only one. Genetics, so sometimes genetic makeup also plays a role on the impact a certain or particular teratogen might have on the developing fetus. This is suggested by fraternal twins that are exposed to the same prenatal environment, but they do not experience the same teratogenic effects, okay? The genetic makeup of the mother can also have an effect. Some mothers may be more resistant to teratogenic effects than others. And just being male or female. So males are more likely to experience damage due to teratogens than females. It's believed that the Y chromosome, which contains fewer genes than the X, may have an impact. So you know that males with an X and a Y sex chromosomes have more sex-related or sex-linked problems such as color blindness, which is not caused by a teratogen, but males just have less protection because they don't have two X chromosomes, okay? But you know, technically only one X chromosome is functional in any cell of your body, whether you're a male or female. If you're a male, the X chromosome, whatever X chromosome you inherited is from your mom, is in all of your cells. In a female cells are a mosaic of either the X chromosome that you inherited from your father, or the X chromosome you inherited from your mother being active, and the other one is inactive, right? So you remember bar bodies, all that good stuff. All right. This, don't start memorizing this, but this just gives you some idea of the harm that can be caused by teratogens and what specifically would be subject to abnormalities. So the dividing zygote implantation and gastrulation, not susceptible to teratogens, because if a teratogen, if a female is a pregnant female is exposed to a teratogen that is harmful to a potential fetus, it would cause death of the developing fetus, okay, of the embryo. All right, but exposure, and again, it depends on the timing of the exposure, the amount of the exposure, the number of teratogens that are potentially part of, or the woman has been subjected to. So here we have the age of the embryo in weeks. Once again, we remember this delineation here between 6 and 7 weeks. This is when the gonads begin to start differentiating. Up until then, there's been no difference between a male and a female fetus. The one thing I want you to point out, so they skip here from 9 weeks to 16 to 20 to full term. Okay, so that's truncated a little bit. The thing to notice here is that the central nervous system can be damaged or become abnormal at any point in prenatal development. Okay, the central nervous system is subject to abnormalities or damage or the result of these teratogens at any point from 2 weeks after conception to full term. All right, but you notice here the heart is essentially stops maturing at certain between 8 and 9 weeks of it's getting bigger, right? It's growing, but as far as the effects of teratogens on the heart, that really can only occur between 3 and 8 weeks. Same thing, approximately with the upper limbs, the palate, the ear, external genitalia, functional. So major morphological abnormalities in the first 7 or 8 weeks, after which, you're not home free, but functional defects and minor morphological abnormalities, okay? So this too, I'm just gonna end up here today. This gives you another idea of when certain features of the developing fetus are most susceptible to teratogenic effects. And if you want a little bit more detail, which I'm not going to go into, but I want to provide this information for you, you can take a look at these different aspects. Like if you're interested in what kind of teratogenic effects could affect the kidneys, this gives you a little bit more information here, okay? The eyes, lungs, respiratory abnormalities, etc. So most of these, notice, are very, very early in embryonic and fetal development, sometimes before a pregnant person is even aware that they are pregnant, okay? So this is something important if a person is trying to become pregnant, to make sure even before pregnancy is detected that you try to minimize exposure to these teratogens, etc. Okay, so next time we're going to start talking about some of these teratogenic agents in more detail. All right, starting with physical agents, maternal health factors, environmental chemicals. This is a very important and very scary one. Infectious agents. This is where I'm going to try this weekend to catch up on the latest COVID information regarding pregnant women. They've had almost 3 years to do studies of the COVID virus, and I'm hoping that there's more consistency in the within the medical community about COVID this semester than there has been for the last 3 years. Because I've been trying to stay up with the information, and a lot of it was, well, we think this, but maybe that. And so hopefully there's a little bit more factual information out there that is consistent with what most of the medical community is producing. All right. And then, of course, drugs, recreational, over-the-counter drugs, etc. Okay, well, have a great weekend. Don't forget to take the quiz, and we will see you back here bright and early Monday morning. You know what? I don't think any of the sound was recorded. So you all are going to be very, very popular over the next few weeks. But I think a couple of videos are pretty useful. I got an email asking if I have the handheld microphone, and I'm like, I didn't even touch that thing. What are you talking about? They said, it's missing. Yeah, you're welcome. Have a good weekend. Oh, don't I ever cool down. Heat can be a teratogenic agent. Just saying.